the brain is the tissue that consumes oxygen faster and more and and requires it more than any other tissue by far. So you do this to a grandpa, he comes out of the adventure with dementia.
Dear God.
Yeah. Well, he belongs to God at that point.
Wow.
Our neighbor's choice.
Well, we're here in treat for a treat today. We've got my big fat gas panel today. It's because we're talking about hypoxia, oxygen, carbon dioxide, and the role that all of this balance between oxygen and carbon dioxide is something I've been fascinated with ever since Dr. Ray Peat got me interested in the topic. And along the way, these are some of my friends that are talking about these various issues related to hypoxia and how to, Brad Marshall, our guest here, he focuses on the role of PUFAs, polyunsaturated fatty acids, also found primarily in seed oils and other industrial fats that are rancid and toxic. And he's focusing on how that affects the levels of hypoxia in our bodies. We can get into that at a deeper level with him. We have Steve Scott, who's the founder of Carbon Genetics, where he's using CO2 therapy, whether it's in the form of inhaling it or through the process of absorbing it through the bodysuit. I've enjoyed both methods and can attest that they are quite relaxing, and I think they're in store for a lot of transformative health effects for myself and improvements. Some world-class, world champion athletes use his devices for CO2 and have amazing ecstatic victories. And then we have the illustrious Dr. Coleman, who is the author of 50 Years Lost in Medical Advance, and also someone who is quite published, having completed the work of Hans Selye's The Quest for the Mammalian Stress Mechanism. He's an anesthesiologist and someone that we enjoy hearing from. So thank you all for coming on the program.
Well,
Thanks for
thanks for
having us.
having us. Yeah.
Yeah. Now, Steve, are you doing the bodysuit right now, or because you look really tranquil?
I am not doing it now.
I thought you had a sleeker black unit that I didn't know about yet. My goodness.
Yeah.
Wow. Well, Dr. Lewis Coleman, let's start off with you since you're our doctor in the house. Why do we need to understand oxygen and carbon dioxide and how that affects so many of the diseases that we have today, right? Can you give us a
Okay.
little lay of the land of that?
Sure. Well, the main focus of this would be the what you call the mechanism of oxygen transport and delivery, which captures oxygen from the atmosphere and transports it to cells deep within the body because obviously every cell in the body requires oxygen for its metabolism, and the main metabolic pathway of all eukaryotic cells that make up all your multicellular animals and plants that live on the surface at least of the earth use what's called the Krebs cycle as its main metabolic pathway. And I'm sure Brad has a lot to add to this, but I'm kind of interested to see what he has to say about the cellular metabolic pathways that I'm not an expert on. But I do know from my basic biology in college that the Krebs cycle is the primary metabolic pathway that generates what you call ATP, or adenosine triphosphate. Um, think of that as like little cans of gasoline or oil that float around in your blood and your tissues, and the cells use that as to power all their activities. It's I call it the universal, what do I call it? The universal source of cell and extracellular enzyme energy.
Mhm.
Okay, so now the mechanism of oxygen transport and delivery is amazingly misunderstood and is not taught properly. And this is an accident, in my opinion. It's actually been known for more than 100 years. It was discovered and characterized around the turn of the previous century, um, where they studied how hemoglobin binds to oxygen. They call it the hemoglobin oxygen dissociation curve. And I'm blanking. I'm trying to think of the name of the Danish researcher who was famous for this. But the important thing to understand
Is
is
it
that
Bohr?
who?
Bohr is his last name?
Yeah, Bohr. Was it Niels Bohr? But his
I
name
can't
is
think of his
Bohr.
first name now.
Yeah,
Niels Bohr.
and
Niels Bohr, yeah.
yeah. There's another Bohr who came along later who was prominent in theory, but no, this Bohr was famous in his own time, and so he discovered this oxygen hemoglobin dissociation curve, and that you need to understand that to understand how the oxygen transport mechanism works. So hemoglobin binds avidly to oxygen. It doesn't want to let go. And so when blood is passing through the lungs, the purpose of breathing is misunderstood. Everybody thinks that the purpose of breathing is to first and foremost bring fresh air into the lungs, which of course it does, and then get rid of carbon dioxide, which it does, because carbon dioxide has been vilified as toxic waste like urine. So the idea is that you got to get rid of it all the time. Actually, what's going on is carbon dioxide interferes with the binding of oxygen to the hemoglobin molecule. So what's going on here primarily is when you breathe, you're lowering the level of carbon dioxide in the lungs, so that that exaggerates the binding of oxygen to the hemoglobin molecule as blood passes through the lungs. And this is sort of, you can think of it as a safety reserve or something that makes sure that oxygen or the hemoglobin is 100% saturated as it passes through the lungs. Now, each hemoglobin molecule has 4 binding sites. And the truth of the matter is that anybody who's halfway healthy, or even when you're sick, your hemoglobin emerges from the lungs 100% saturated with oxygen. In other words, it binds to oxygen so avidly that it's not a problem saturating hemoglobin with oxygen. And the first lesson here that nobody understands, doctors, nurses, you name it, you don't need to breathe oxygen. The first thing they do when you get in near an ambulance or a hospital or any kind of medical personnel, they grab you and whack, they slap a mask on your face and give you 100% oxygen. It does no good. It's ridiculous. It's like a joke, because if you just breathe God's air, you're going to saturate your hemoglobin and with 100% with oxygen. And the hemoglobin carries 99% of the oxygen in blood, and less than 1%, I think, I'm rusty on this, but a very tiny portion of the oxygen in blood is dissolved plasma, but it's insignificant. It's meaningless. So already you see one of the fallacies of medical practice is this mania for making everybody breathe 100% oxygen. Well, actually, it's harmful, because oxygen is extremely reactive. It's toxic. And if you say, there's baboon studies, as we know best, if you have the baboon breathe 100% oxygen, for more than 24 hours or so, you can actually kill it. It'll literally burn the lung tissue.
Mm.
So this is not good for you. And furthermore, it's a fire hazard. Anytime you start horsing around with oxygen and a mask, and everybody, I mean, the nurses are just stubborn as can be. They will not listen to you. They'll get mad at you. Hey, take the oxygen off or turn it down. They've got it up to 10 liters or something, and the oxygen's flying all over the place. And so you get these oxygen fires in the hospitals. And I mean, they really hurt people. It's funny. And they don't understand that it's not serving any purpose whatsoever. Okay. So then the heart pumps blood, from the lungs. It goes to the left side of the heart, and the left side of the heart, of course, is a lot bigger and stronger than the right side of the heart, and it pumps blood to the rest of the body. Now, here comes the next misunderstanding about this mechanism. Everybody thinks that blood pressure is the driving force of blood flow. No, they think that blood flow characteristics are identical water, but water and blood are profoundly different. Water is what you call a Newtonian fluid. What that means is when you accelerate water in a pipe or an artery, it causes exponential increases in blood turbulence and turbulent flow resistance. And the faster you accelerate the water in the pipe or the artery, the greater the flow resistance that it exerts. It forms these turbulent flow patterns with the forward moves along the inner wall of the pipe. And those curve around and they push the slower moving elements of the water to the center of the pipe. And then it goes backwards. And the harder you push, the more, flow resistance you get. That's why it takes so long to fill a swimming pool or even a bathtub. You can only put so much water pressure on a municipal water system. And then if you get beyond, the turbulence becomes a practical problem. I hope I'm making myself clear. So blood contains particulates, namely the red blood cells, which are unique in vertebrates. I mean, all vertebrates have red blood cells. The hemoglobin is always enclosed in a cell, a red blood cell, and like birds have a certain shape and size and buoyancy to their red blood cells. It's different from amphibians and ours, or what you would think of as crocodiles. Every different class of vertebrates has a different type of blood cell. They can be identified by their blood cells. So the red cells of mammals are tiny, way smaller than the blood cells of any other mammal or any other vertebrate. I'm sorry, and they're biconcave. They call them scaphoid. So when blood is accelerated in an artery, then all the red cells spontaneously form what we call stacks. They form little, towers or stacks. The French call this rouleau. And so those stacks, as they form, they prevent the formation of these turbulent flow patterns. And by the way, this used to be misunderstood until about 20 years ago, and a team of Belgian engineers used laser photography to photograph water flow in pipes, and they literally took laser photographs to prove this, computer-derived theory, mathematical projection of flow turbulence. And so they confirmed they tested and confirmed the theory. says the head researcher's name was Hoff, Hof. And if you want to read a really interesting engineering study, I recommend that. It's really an eye-opener. So when the heart beats, it expels its contents in less than a tenth of a second. It's really amazing. Nobody appreciates this. It just goes, bloop. That's it. And the rest of the time, it beats, 8 a minute, or depending on your health status and so forth. Like a trained athlete, it'll have a resting heart rate below 40 beats per minute. And his heart his heart is so efficient. He's got so many capillaries. His flow resistance is negligible. And so his heart just goes, eh. And when he exercises, his heart rate rises up to 80 maybe, while he's running 90 miles an hour, because he's got exercise conditioning. He's formed extra capillaries in his muscles and tissues. So there's virtually, the faster he runs and the harder he exercises or whatever, this recruits more capillaries and the flow resistance doesn't increase. And your blood flow in your arteries is like superconductivity. It's like the harder the heart pushes the blood, the lower the flow resistance becomes, because the red cells. Am I making myself clear so far?
Mm-hmm.
Okay.
Yep.
All right. If you guys have questions, squeak. All right, and I'll try.
Well, Brad, what are you thinking? How would you like to chime into this so far?
I mean, I'm just curious to hear the rest.
Yeah.
Most of what I focus on is what happens once the oxygen is delivered to the cell. So I haven't thought as much about these components of it. So I'm
Right. Yeah, that's so I was looking forward to hearing what you had to say, but I'm glad to get a chance to paint this picture, because it's not your fault. They don't teach this in medical school. Now they did in my medical school. Let me just make it. They taught it in my medical school, but I think that my medical school was exceptional. Really, I was very fortunate to go there. But at any rate, to go on and complete the picture. So the blood is expelled into the arterial tree. Now here again, everybody in conventional orthodox medicine, again, they're assuming that blood pressure is the driving force. So they think that blood flow is controlled by vasoconstriction and vasodilation, but blood is a non-Newtonian fluid. So when you have narrowing of an artery, all that happens is you speed up the blood flow. Okay, so if an artery contracts, then the blood speeds up. And well, once in a while in the operating room, if you're in there for a long time like I was, 40 years or whatever, once in a while a surgeon makes a mistake and goes oops, and it slices an artery, and you'll see the blood go zip, 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 right? Because the artery is trying to contract and it speeds the blood up, and then it squirts quite a distance, right? It's got it accelerates it by a velocity.
Mm-hmm.
And of course, your simplistic thinking is, oh, well, obviously the heart beats and propels the blood, and that seems obvious, right? But real hemodynamic physiology is backwards from what our common sense tells us. Everything is different. Everything is backwards. So what happens is that when your heart expels this bolus of blood out into the arterial tree, it's muscular, but it's also elastic. And the muscular muscle is different from the other muscles in the body. Like the cardiac muscle has lots and lots of mitochondria in it because it's all the time working. The skeletal muscles are sort of midway in between. They have a fair number of mitochondria, but that produce the ATP energy. And Brad will get into telling us all about that, I'm hoping. And then the vascular muscle has very few mitochondria because it's absorbing its oxygen straight from the arterial blood, which is 100% saturated with oxygen. So it doesn't work very hard. So what happens is the entire vascular tree gently expands to accommodate the bolus of blood that's injected into it by the heart. And it functions as a secondary heart. So the heart's not doing all this work by itself. So now, as soon as the heart has expelled its contents, now everybody thinks that everything in the body is super, super efficient, but muscle isn't. Because when muscle contracts, it's like a ratchet system where you have actin and myosin, moving vis-a-vis each other through little catches or ratchets, and it goes click, 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 like this. And each of those clicks consumes ATP, but then it can't let go unless it has to unclick. And each unclick takes another ATP. Clunk, 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 right? So now the heart itself is beating a lot. It works all through lifetime, right? It's, but it's weak. It doesn't have to work very hard if you're healthy, the Newtonian characteristic of blood. In other words, the harder it beats, the easier it gets to squirt the blood out of itself, okay? So, but then the heart has a cartilaginous skeletal framework so that when it relaxes, the cartilage opens up the heart, or restores the heart's volume, and pulls fresh blood from the atria into the ventricle all at once, without the heart having to consume a lot of extra energy relaxing itself. Okay. So anyway, so now the arterial tree, having been charged with a bolus of blood, it very gradually and gently restores itself to its resting volume. As it does so, it pushes the blood towards the capillary beds. Well, now what are we measuring when we measure blood pressure? Okay, well, here's what happens is the distal third of the aorta is about a third the diameter of the proximal aorta. Oh, I'm running out of battery power on this darn thing. That's what's squeaking. Let me switch. I may have to switch to a different speaker and headphone here. Didn't expect this to be a problem.
I can jump in for a second.
Yeah, why don't you chime in for a second, and while I get this straightened out.
Yeah, so as you were talking, I'm an evolutionary, I think like an evolutionary biologist, right? I like to put things in order of sort of what came first. And what you're describing is very interesting because I know that, or I'm pretty sure that I know that when you look at the chordates are things that have a spine mostly, except for the oldest chordate is a thing called Amphioxus, which is a little looks like a fish, but it's not a fish. It doesn't have a skeleton. And you can still catch these. They're in the shallow bays, and they eat them in Japan. And anyway, what's interesting is the Amphioxus does not yet have a heart, but what it does have is contractile blood vessels to move. It's not really even blood yet, but it uses it to move the kind of interstitial fluid around through the organism. So what you're describing is perfectly logical with how things kind of evolve. The contractile vessels or the contractile arteries came first and the heart came later, probably as a way to help shunt the blood into the contractile arteries.
Yeah, what you're talking about is a different stress mechanism. And so all I'm talking about is exclusively the mammalian stress mechanism, because each vertebrate has a different stress mechanism.
I was just saying that in the chordates, which is things with backbones, animals, or yeah, mammals,
That's what
lizards,
we need
fish.
more of in this country, don't we? People with some backbones, huh?
That's right.
Bring back the
Mammals.
chordates.
Yeah.
Yeah, and so but the amphioxus, the lancelet, is at the base of that tree, that phylogeny. So they have uh, a cord. They have a notochord, which is what eventually became the skeleton, right? Or the spine. But they have these contractile arteries, just as he was describing. And they do not yet have hearts, right? And so the contractile arteries came before the heart actually did, interestingly.
Hmm.
And insects is another example. I mean, we're talking about different stress every time we do a shift like this. You know, the
Yeah,
chordates is one thing, they have their stress mechanism. Insects have another stress mechanism. So insects inhale air through their abdomen through sphericals. And so if you look at them carefully, you'll see their abdomen is expanding and contracting. And it's a very, very effective stress mechanism, but probably is only good up to a certain size. And then you start running into size problems.
yeah,
Um, Now, the only what I've really done is I've identified the first stress mechanism to ever be described, and that really is courtesy of um, thousands and thousands of research studies that have been performed on mammals because everybody's interested in medicine and the government subsidies pay for that. They don't pay for the biologists to do this kind of intense investigation. Um, and of course, then there's plant stress mechanisms too. You know, the big classes of plant gymnosperms and angiosperms that we see on the surface of the earth. That's the most common two categories. But then I don't want to go too much further here because I'm out of my water. Real son could, I'm trying to get him. He's going to get his PhD here in a couple of months, and then I'm wanting him to see if he can describe one of the stress mechanisms for plants. But that's a different ball of wax. That's plants are way complicated. But anyway, to finish off where I was, because um, the blood, by the time it gets to your capillaries, the flow is relatively constant and uniform. Okay, and so your blood perfuses through the capillary beds, and your cells are constantly generating carbon dioxide. So the slightly higher level of carbon dioxide in your organs and tissues loosens the binding of oxygen to the hemoglobin molecule and releases it into the tissues. Now, you know, I proved this in the operating room for myself, although there's plenty of other studies that have done it too. Um, when, like in the operating room, what I did was I uh, gave the patient a lot of narcotic, and narcotics reduce respiratory drive. And so then the CO2 level within the body would build up. And the partial pressure of the carbon dioxide normally in the body is about 40 torr, okay, 40 psi. But I would let it go all the way up to well above 100, shall we say. I don't want to scare anybody, but it's totally harmless. Now, there's dog studies. If the only way you can get in trouble with carbon dioxide is if it gets up well above uh, 200 torr, at which point you're flirting with um, the carbon dioxide will start to interfere with the binding of oxygen in the lung, okay? Because it inhibits the binding to the point that you start to um, have your arterial blood's not saturated anymore. And I hope I'm making myself clear with this. I didn't get anywhere near that in the operating room. And besides, nowadays we have what you call pulse oximeters. Now, this is a whole different lecture. It's not the way this thing works. Have you guys all heard of a pulse oximeter? Know what I'm talking about?
Yeah.
Mhm.
Yeah.
You all do? Yes, yes, yes.
Yeah.
Okay.
Mhm.
Yeah.
All right. Well, the pulse oximeter shines three different wavelengths of light on your skin, and it bounces those wavelengths off the hemoglobin molecule, and then it has a photonic receiver, and and then they have an algorithm that analyzes the relative reflectance of those three different wavelengths of light and tells you whether your hemoglobin is saturated or not. Well, now it gets kind of complicated, but basically, when the when the the algorithm says that it's got 60% saturation, it's the hemoglobin is actually 100% saturated or close to 100% saturated, because what it's looking at also is the oxygen saturated in the plasma. So, if you just run the patient with a pulse oximeter reading 60, he's fine, actually, but you can't tell that to a nurse. Oh, but what's going on there in the operating room, in 99.999% of the operating rooms throughout the world, anesthesiologists have been brainwashed to think carbon dioxide's bad, and mechanical hyperventilation is the answer. Okay, you gotta breathe for that patient, by God, and more breathing is better, no matter what. More, more, more. Okay, so they hyperventilate the daylights out of the patient. They deplete the carbon dioxide from the patient's body. And the pulse oximeter is way happy because now you've exaggerated the binding of oxygen to the hemoglobin molecule. But on top of that, you've supersaturated the plasma. And the pulse oximeter goes, ooh, it runs right up to 100. And all your nurses go, wee, good doctor, right? But what's happening is that the body is depleted of carbon dioxide, so it's not releasing oxygen into the tissues. And now, the brain is the tissue that consumes oxygen faster and more, and requires it more than any other tissue by far. So you do this to a grandpa, he comes out of the adventure with dementia.
Dear God.
Yeah. Well, he belongs to God at that point.
Wow.
And this happens all the time. And everybody goes, why is that? How come? It's a big mystery, don't you know? My God, we've understood this for more than 100 years. This is basic physiology. It's like they control medical education and textbooks and
Yeah.
journals and medical boards. And it's like the tail wagging the dog all over the place. So medical students are brainwashed. They don't even know it. And then you go into anesthesia school and you're brainwashed 10 times over. And you cannot change these people. Doctors are not scientists. They're doing this fundamental psychological behavior. They got to be like everybody else. They want like lemmings jumping over a cliff, or penguins in a herd or a flock. Everybody wants to be all cozy and quacking alike.
Would,
Quack,
would
quack,
not,
quack.
but Dr.
Okay?
Coleman, then not only do they cause dementia, they also give people diabetes when they go to the surgery practice, right?
Yes.
How's
Well,
that
yeah,
happen?
because let's see, how would that happen? Well, I hadn't ever been asked that question before. Um, I can tell you that carbon dioxide not only loosens the binding of oxygen to hemoglobin and releases oxygen from the hemoglobin into blood, but it's the sole source of respiratory drive, what makes you breathe. And it directly releases nitric oxide from the vascular endothelium. Now, your capillaries have only one layer of cells, and that's pure vascular endothelium. And the vascular endothelium is this delicate layer, one cell thick, on the inner surface of all your larger blood vessels. So actually, the vascular endothelium is the heart and soul and focus of the stress mechanism, the mammalian stress mechanism, which I won't go into right this moment. But carbon dioxide is the primary regulator of the capillary gate mechanism that operates in capillaries that regulates the flow resistance in your organs and tissues. So carbon dioxide releases nitric oxide. Now, the nitric oxide binds to calcium, reduces thrombin activity, and I'll cut this whole, but in the research literature, this is called nitrogen neurogenic vasodilation. Sorry, I have to say that because that's what it's called. But it doesn't cause vasodilation like opening up of the blood vessels or anything like everybody thinks. See, because everybody's got this stubborn, entrenched belief
Yeah.
that the blood vessels muscularly contract and then relax. So they think this must be vasodilation, but the way it works at all. It's actually what's happening is you're generating a protein called insoluble fibrin in the capillaries that forms little strands, and the strands are generated by sympathetic nervous activity. And so they form in the capillaries and they increase flow resistance. They don't close the capillary per se. We can speak of closing the capillary gate or opening the capillary gate, but you're not literally shutting off flow in the capillaries. You're just slowing it down. Okay. And then when you get rid of the insoluble fibrin, and it will spontaneously self-destruct because it contains a link in its molecular structure called plasminogen. And the plasminogen breaks down into an enzyme called plasmin, that attacks the insoluble fibrin structure and disintegrates it into what you call fibrin split products. That opens the capillary gate. Making myself clear?
Yeah.
Or
Brad,
everybody
do you
asleep?
understand?
I mean, yes, I understand that. So this is the process that instead of the artery or the capillaries, I mean, physically constricting, instead it's adding material inside, which increases resistance to flow
There you go. Okay, good.
via a mechanism entirely different than literal
Excellent. Excellent.
dilation and undilation.
Okay,
Don't
excellent,
the
Brad.
don't the blood vessels higher up in the tree, don't they have smooth muscles that
Yes, but like I said, even if they do contract, and you can make them contract with electricity or something like that and certain
Yeah.
chemicals, but all these studies are done in vitro, outside the body. Okay, and they take a section of artery and they put it in a salt bath or something, and then they goose it with electricity or this and that and the other chemical, and yeah, it's muscle, and the muscle contracts and the lumen gets smaller. That's all true. But
Mm.
in real life, in the body, if you actually constrict the diameter of the blood vessel, what happens is it just speeds up the blood. Doesn't do any good.
Mhm. Because
It's,
normally when you like breathe carbon dioxide, there's been studies showing that it increases blood flow to the brain by 50%, and it was attribute that to vasodilation. But you're saying it's
right.
actually vasoconstriction that's increasing the blood flow, blood velocity, blood volume to the
No,
brain.
no, there's no vasoconstriction. There's no vasodilation going on, except that when the arterial tree is expanded by that bolus of blood, then it slowly contracts, and the muscles, you know, gent not very strong muscles. They can't do much work because they don't have any mitochondria. So, they slowly it slowly restores its resting volume, and that's not it's like a very gentle force. I
I mean,
really
I feel like we're talking about the same thing. It's just you're arguing that the mechanism by which the blood flow is increased or reduced is different than is commonly believed, right? I mean,
correct,
you're
correct.
you're essentially in principle, the constriction is happens, but it's happening by a mechanism different than literal physical constriction.
Right. And I'm hoping I can persuade you to read my book and critique it because it's got a chapter in there on the unified theory of biology, and that's what I want you. I've been looking for a biologist like you
Cool.
who can appreciate this and evaluate it for me.
Okay.
Uh,
Yeah.
I think just from what you've said, you're going to really enjoy reading this. It's going to give a radically different view of anatomy, taxonomy, behavior, um, speciation, evolution. It's going to resolve the disparities of Lamarck and Darwin and Baldwin and saltation. Um,
Okay.
these are all major mysteries, right? In biology.
What's the name of the book? Sorry, this
Uh,
just so people know, this conference came together this afternoon, so we haven't had a lot of time to do background research.
the,
Oh, right. Yes, you said that. Yeah.
uh, yeah, I'll get you a PDF copy by email, or I'll send you a
Okay.
link so you can get it, download it from my Dropbox,
Sure.
if that is adequate.
That should be fine. Yeah.
Well,
Yeah,
Brad Marshall's been looking into this concept of how oxygen relates to obesity and these hypoxic states that people are in when they're morbidly obese. And I wanted to bring Steve Scott on because he's into researching CO2 and its relationship to oxygen and healing, people's problems with obesity and all kinds of other metabolic dysfunctions. And I thought you guys could weigh in together. So just jump in and talk about it. I don't want to get in the way.
Yeah, I'd
well,
love to hear your idea about that.
let Steve have at it for a bit
Yeah.
here.
But I'll let Brad go talk about his ideas
Yeah.
about obesity and hypoxia and whatnot.
Yeah, okay.
Well, so right. So I look at the, I look at inside of the cell what's happening, right? And this is, in my opinion, that this interview is actually very well timed because I'm literally writing. My plan tomorrow morning is to write this article and post it on my blog. So probably that'll maybe be out before this is. I don't know. It'll come out together. And the idea is that what I've seen is that once oxygen is actually delivered into the cell, one of these other big misunderstandings, like you've mentioned, when you take biology 101, they show you this drawing of a cell, and the cell has like a nucleus, and then around the nucleus is the endoplasmic reticulum, and over here there's a big old mitochondria. That's not reality. The reality is the mitochondria are very small and they're embedded into the endoplasmic reticulum, which is all around the nucleus, right? And so So you have the nucleus, and around that, there's these layers of endoplasmic reticulum, and there's layers of mitochondria buried in there. And so once the oxygen gets into the cell, it now has to migrate through the endoplasmic reticulum to get to the mitochondria. Now we have a system of enzymes. One is called delta 6 desaturase. There's a delta 5 desaturase. There's this delta 9 desaturase. There's cytochrome P450 enzymes, and there are cyclooxygenase enzymes. I might be wrong about the cyclooxygenase enzymes, but the other ones I'm sure of, they live in the endoplasmic reticulum. And so when any of those enzymes are elevated, that predicts your likelihood of developing diabetes over time. That predicts they're strongly correlated with, degree of obesity, degree of metabolic syndrome. To me, that system looks like those enzymes are there to prevent the oxygen from getting through the endoplasmic reticulum to the mitochondria. They're essentially an oxygen shield. And why does it work that way? Because when a bear wants to fatten up for winter, all they have to do is increase the expression of these desaturase enzymes, and that will fundamentally flip the adipose cell from being primarily oxidative to being primarily glycolytic. Right? It's a pseudo hypoxia. The problem is not always that. I mean, there are problems with oxygen delivery. It's not that they're not, but there's this further problem once the oxygen gets into the cell, that if your desaturase enzyme system is cranked up, that oxygen is essentially being intercepted as it moves through the endoplasmic reticulum. And the primary fuel of all these systems are well, at least the delta 6 desaturase and the delta 5 desaturase and the cyclooxygenase system is linoleic acid. The cells prefer to put linoleic acid into the SN2 position of the membranes, and the endoplasmic reticulum is a membrane, right? It's a membrane that lives inside of the cell. And so the endoplasmic reticulum is rife with linoleic acid, and the role of it is when the bear decides it needs to fatten up for winter, you've got the fuel right there along with these enzymes. Delta 6 desaturase. The linoleic acid, by the way, is constantly being released from the membranes and then being reacylated in this cycle called the Land cycle. And so the cells take the linoleic acid. Delta 6 desaturase puts a double bond in it that consumes a molecule of oxygen. Then another molecule sticks an acetyl-CoA group on it, and another enzyme, D5D, desaturates it a second time. That consumes another molecule of oxygen. And then a cytochrome P450 enzyme sticks another oxygen on the end, making it 20 heat at that point, right? And that consumes a third molecule of oxygen. And now the 20 heat is exported via transporters in the cell into the blood, and it's transported, and it can be burned in the mitochondria, or it can be urinated out. But the point is that linoleic acid was essentially used as a fermentation process to soak up the incoming oxygen so that it's not getting to the mitochondria, and exported from the cell. That is my hypothesis of how the system works and why these desaturase enzymes are elevated if winter is coming in hibernating animals, and they're elevated in obesity and diabetes. I believe that
is that a purposeful thing, or is that just a result of having too much linoleic acid or something like that, or is it intentional?
Well, So I think it's a lot of things. I think having a lot of linoleic acid is of course providing fuel for the system, right? What you actually see, and this right. So one of the paradoxes that we see is that um, so a lot of us think that we've been consuming too much linoleic acid, and linoleic acid levels have increased over time, right? So you're providing more fuel for the system. However, if you look at population level studies, if you assume that everyone is eating the same amount of linoleic acid on average in the U.S. population, which is probably more or less true, um, the ones who have the lowest level of linoleic acid in their adipose tissues are actually the most likely to develop uh, diabetes, and some of this obesity. And so I think what's happening is that in those people, your adipocyte, because remember, once this 20 heat is produced and ejected from the cell, that cell can just replace it from linoleic acid in the bloodstream, free fatty acids, right? The
Hmm.
adipose tissues are constantly releasing these free fatty acids, and the adipose tissue can constantly take them up, suck them into their endoplasmic reticulum membranes and use them as part of this ferment. I call it an oxidative fermentation, right? And so now your adipose tissues, instead of being oxidative and using their mitochondria to do oxidative phosphorylation, now they're fundamentally glycolytic. And the fuel for them to remain glycolytic is the linoleic acid. However, all of this is controlled by enzymes, right?
Hmm.
And so if you don't have elevated delta 6 desaturase, delta 9 desaturase, and delta 5 desaturase, you won't necessarily have this problem. And in my opinion, that explains why different genetics are more prone to, different, there are known polymorphisms in these genes that make them more or less active. And if you have the active version of delta 6 desaturase, that predicts that you will be more likely to become obese and or diabetic. And so I think that's just controlling this process of fermentation. And then just to bring that method home, when you look at an obese person, they put catheters into the capillary beds.
Hmm.
You can see that the fat cells are not using oxygen. They're using very little oxygen, and in fact, compared to the rest of the body, right around the adipose tissue is actually higher in dissolved oxygen um, than the rest of the body. So the oxygen is getting there, but it's not getting in, at least in the adipose tissue. Now that situation might be very different in muscle and in liver, but specifically in the adipose tissue, the adipose use these enzymes, I believe, to become hypoxic. And you have this irony there's an enzyme called HIF1, which is an oxygen sensor that's in all the cells. And when cells become hypoxic, HIF1 or the enzymes that are controlled by the transcription factor HIF1 are increased. That tells you that the HIF1 has been activated. And in obese tissues, you see this ironic situation where the uh, oxygen pressure in the tissue is not necessarily bad, but yet the oxygen or the cells have this activated HIF1, a sign of hypoxia. It's a pseudo hypoxia because the oxygen is there, but it can't get to the mitochondria, and therefore the cell is actually in oxygen stress.
I
Mm.
do have a question.
Sure.
Not to interrupt you, but I've been wanting to ask this. I mean, this is very interesting what you're saying. When I was in medical school, which is like I'm a dinosaur, right? Uh, but I remember they taught us that there's three essential fatty acids
Right.
in your diet: linoleic, linolenic, and arachidonic acid.
Right.
And they didn't have any idea like what you're talking about back then. I mean, you're got all these modern studies with newer research techniques and everything. But what about the linoleic and arachidonic acids? What are they doing?
Well, so it's kind of the same, especially I think the two that are most responsible for this are the linoleic and the linolenic acids. Both of those are in a position where they they can consume the most oxygen because to go from the process of going from linoleic to arachidonic acid consumes two molecules of
Oh,
oxygen.
can they be converted back and forth?
Yes. So the
Ah,
that's right. Right. So after delta 6D and delta 5 D6D and D5D act, now it's arachidonic acid. Arachidonic acid is the thing that yet another oxygen is used to convert it to 20 heat.
uh-huh.
And that's the thing that gets eliminated in the urine or can go into the
Right.
mitochondria as well.
See, I don't think they
Um,
knew that back in my day.
well, I think I mean, I think they may not have known that. I think, yeah, I don't know when the dynamics of those enzymes were figured out. Probably
Right.
later, but I think it's only, well, I guess it's only me that sort of put together that what the idea that this system is essentially acting as an oxygen. Everybody looks at it's similar to what you're talking about, where like we see all the clues, but you're looking at the problem wrong, right? What we see is we see all these people say, oh, it's these oxidized lipids that are causing all of these problems. It's the 20
Mm,
heat that's causing we see the 20 heat rise and it's elevated in obesity. The 20 heat is causing the problem. I'm saying that the 20 heat is a just a symptom of the problem. The problem is the fact that these enzymes are consuming oxygen in the endoplasmic reticulum and not allowing it into the mitochondria.
hmm.
And
Well,
evolutionarily for mammals, this is an advantage because if you live in a northern climate where you need to hibernate half of the year, then this is a really useful system,
hmm.
right? And so what I go and so this is a good analogy and why I think about evolution, right? So in my current blog series, I talk about copepods. Copepods are like tiny shrimp. They're little crustaceans, and they live in
Right.
the ocean. They're basically the basis of the marine food web, and they eat algae, and they in turn get eaten by like bait fish, right? And so, and when you look at the copepod, what they do is if you feed them a type of algae that has linoleic acid and linolenic acid, they just vaporize that. And they'll hang on to some of the like the DHA and the EPA, the longer chain, uh, you know, omega 6 and 3 fatty acids. But the linoleic acid and the linolenic acid go just like that. And furthermore, what they do is very interesting. Is the female copepods when they're actively producing eggs and they're very anabolic, they swim up to the surface during the day, or sorry, during the evening. They feast on algae, and then they swim back down to the oxygen minimum zone where dissolved oxygen is very low. And my argument is that this makes them very anabolic and glycolytic, and that they're using that linoleic acid and the linolenic acid as the fuel for this oxidative fermentation. Whatever oxygen is there, they're using for this fermentation, and that actually clears NADH from the cell, and it allows them continue to do glycolysis because we have this long-standing problem in all cells that when you do glycolysis, when you use glucose for fuel, you create NADH. And uh, NAD takes a pair of electrons from glucose or from the glucose byproducts, and now those electrons have to go somewhere. And so a lot of things have solved this by fermenting the pyruvate created from glycolysis back to lactate, and they spit out the lactate. And that's how you make sauerkraut, right? The bugs in the sauerkraut are taking glucose out of the sauerkraut. They're doing glycolysis to generate ATP. Then they're ejecting the carbon framework of that glucose as lactic acid, and the sauerkraut becomes acidic. And other animals have solved this through uh, fermenting alcohol, such as yeast. But in a multi-celled animal, you need a different solution, right? You can't, all of the cells can't be ejecting lactic acid into the interstitial fluids, or you're going to die of lactic acidosis. Similar argument for ethanol, right? And butyrate isn't great to do that with either. And so the animals had to come up as they became multicellular, had to come up with a solution to this problem. And I believe their solution was ferment the PUFA. That's the thing that you can ferment and eject, and you won't die of acute toxicity, right? And so, and the sort of evidence of this is if you look in the medical journals, they say, well, 20 heat is an important signaling molecule that's used for angiogenesis, which is when cells want to build more capillaries. And
Mhm.
of course, that's true. Of course, that's true. But the copepods are also fermenting the linoleic acid and the linolenic acid, and they're making these oxidized lipids, and they don't have blood vessels yet, right? And so that system predates blood vessels, and so that role of these oxylipins signaling and stimulating blood vessel growth clearly came later, right? The oxidative fermentation is the fundamental underlying process that was there first. The animals, the clever animals, as I like to say on the blog, use that as a signal. Oh, we need oxygen here. We need to introduce blood vessels. But I believe that happened probably 100 million years later,
Mhm.
hundreds of
Mhm.
million years later. Um,
I'm gonna really, I mean, I hope that we can uh, have an ongoing dialogue because
yeah,
you obviously know way, way more about intracellular metabolic pathways than I do. I mean, this is all new to me. It's fascinating. I remember I blundered into a chart once that indicated that you guys, the researchers have identified hundreds of metabolic pathways um, inside the cells, and my take is that there's these athletes who are involved in extreme uh, exercise conditioning, like cyclists, like Lance Armstrong,
yeah,
what Armstrong, and forget there was a guy, an American before him, who was kind of famous as the first American to win that big bicycle race in France, um,
yeah,
and Greg LeMond. Now,
yeah.
Greg LeMond apparently, and this was in Scientific American many years ago, apparently he overexercised and burned out his mitochondria, and he had to quit competing. And so he actually had a muscle biopsy taken, and they've discovered that his cellular mitochondrial level was abnormally low, and so he just had to retire. Um, Lance
Interesting.
Armstrong was also the subject of a Scientific American article. This is when I was in high school, okay? This
Yeah,
ancient stuff. Um, I mean, the guy was a real serious athlete, and so he was participating with researchers to study exercise conditioning. And um, he was just way into it. I mean, he didn't win these races just by cheating. He was really, really
oh yeah.
well conditioned. Um, anyway, I mean, we could go at this for a long time, but um,
I want to add something to this here.
yeah,
So
go
you,
ahead.
so how does that line up with you know, your view of the stress states like obesity and cancer and these other states and relationship to these gases like oxygen and carbon
Sure,
dioxide? You talked
sure.
about to me that you could potentially treat something like obesity with just giving people CO2 every night when they're sleeping, you know? So how would that work? What's the mechanism without, going too deep into it, but just briefly, please?
Well, obesity is a disease unto itself from the point of view of stress theory. Um, I think what's going on is that as you become so heavy, that interferes with capillary flow in your tissues because the tissues are just like I know certainly in the case of the chest, the weight of the fat on the chest wall presses down and interferes with blood flow actually going through the lungs. Um, Now, I don't know enough about the intracellular biology to talk intelligently about that. So
But
Uh,
this is what I like. I like to combine different fields of interest and see what happens
oh
in the
yeah,
process.
this is really good because this is where you tend to start to get serious uh, insights or epiphanies, by
Mhm.
going
Yeah,
into other people's fields, and then you discover relationships like this. So I think that I'm really tickled pink by meeting Brad. I mean,
yeah.
it's
I mean, what I always say is that biology is feedback loops fighting feedback loops,
yes,
right? And so what we're talking about is a positive feedback loop that right the cells are glycolytic, the fat cells start to expand, that puts pressure on the capillaries, that reduces blood flow even more, that causes the adipocytes to become even more glycolytic, and the system becomes
yeah,
self-reinforcing because that
yes, yes,
right
yes,
because
yes, yes,
and that is what and that's what controls the state, right? The state of the organism. What state are we in? Are we an organism that fundamentally believes that winter is right around the corner and we should be conserving fuel, or are we fundamentally an animal that believes that it lives in the tropics in eternal sunshine and there's no need to do that,
right.
right?
Well, now, uh, insofar as just my work on this mammalian stress mechanism, I'm particularly excited right now because when I wrote the book, the one great, question mark I had was what causes chronic illnesses. I have the stress mechanism provides a very simple, straightforward explanation of cancer and critical illness and diabetes, by the way, type 1 and type 2. Um, but actually, um, what I knew at the time of writing the book was that amyloidosis was the theory, seemed to be the cause of type 1 and type 2 diabetes because um, it turns out when I did further work after I finished publishing the book, I mean, I just keep learning more and more. But I did a review finally of I just started looking like you have all these different diseases that are supposedly unrelated to one another, right? That's your
Right,
orthodox medical theory, is that
right,
all the
yeah,
diseases are unique, none of them are related to any of the others, right? Stress theory
yeah.
is saying no, it's hyperactivity of this one that's causing all these seemingly different diseases. Um, and after you've read the book, you'll understand this better, how a single mechanism can actually do that. Okay. I mean,
Yeah, I mean that's very much in line with my thinking, and this is
yeah,
all essentially one fundamental underlying process.
right. And that's where we need to have an ongoing conversation here. Um, but the since I published the book, I had this, what to me is like a breakthrough. I'm pretty excited about this. That I mean, the I didn't understand what caused chronic illnesses, all these rheumatoid diseases, and nowadays they have Orthodox medicine has come to classify uh, atherosclerosis, diabetes, type 1 and type 2, and hypertension, essential hypertension as um, uh, chronic illnesses or rheumatoid diseases. Okay.
Right.
And now a lot of people want to pin everything, pin their tail on the donkey of the immune mechanism and say, well, it's autoimmune disease and it's the immune mechanism is attacking the body in some way or other. That there's no convincing evidence to support that idea.
I I
But
100% agree with you.
yeah, but everybody wants to hang their hat on it or pin their donkey tail on it. And I'm actually writing a paper right now, and so when you read my book, I want you to bear in mind that there's another great. It's like when I was in school, they talked about the plants and but now they're looking at the fungi. There's a whole, and then they're also looking at the archaea cells, right?
Yeah.
Those were completely unknown, and the fungi are now appreciated as like the third great, you know, uh,
Kingdom.
kingdom. There you go. I mean, I had a taxonomy class at Ohio
Yeah,
State.
the mushroom kingdom is rising.
Yes,
Yeah.
it's going to get us here in Bakersfield. It's serious because you get coccidioidomycosis, and if you get around a construction site in a windy day, a friend of mine got this. It damn near killed him. It's really really dangerous.
Dr. Coleman, doesn't that stress mechanism that you discovered have to do with the von Willebrand factor and how that
Oh
works?
yeah, this is the way I got started. Yeah,
VWF factor, right?
factor eight. Yeah, this is the way I got started on this. I went to an anesthesiology conference, Ted Stanley's annual, skiing conference. This was after I actually retired for about 3 years because I got so fed up and disgusted with the politics of medicine, and
Yeah.
so I invested in trust deeds. And so anyway, so I was retired for about 3 years, but then I made the mistake of fiddling and diddling with the stock market and had two daughters in college, and so I went back to work. So I thought, well, I'll get some points and I'll go. And I wanted to talk to Ted Stanley about my anesthesia technique. So anyway, I go to this conference, and the first lecture was by this lady. I've yet to meet her, but my plane was late, or the plane connection couldn't get me to Salt Lake City in time to get to the conference for that lecture. But I got the book, and
Yeah.
that evening I didn't have anything to do. I was all by myself. I couldn't go skiing because it was dark. So I started reading the book. First, chapter in the book was about recent findings in coagulation. And I found out from reading that the vascular endothelium produces von Willebrand factor, which hadn't been known when I was in medical, and it releases it into blood in accord with sympathetic nervous activity. And that wasn't known when I was in medical school, right? I see, Brad, I went to this school that had retained uh, um, a guy named Rodin, who was a Swedish researcher who was very famous. And he had been retained by the school to upgrade the curriculum, which he did. So I got the benefit of his upgraded curriculum, the
Right. Right.
biochemistry and everything. And then after he deliberately waited, I think, until we had been taught the clinical physiology, the vasoconstriction and the vasodilation, all that stuff.
Right.
And then he gave us a series of stress theory lectures that just demolished what we had been taught, we passed the test,
Yeah. That's fun.
jumped through that flaming hula hoop. And then this was like, we weren't tested on this. He just gave these lectures. He gave these lectures to like 5,500 medical students in his life. And so it was amazing because like this, I woke up first time at, in medical
Yeah.
school. I'm in the back row. I'm a rotten student. The whole thing is just a big memorization party, which I hate memorizing,
Yeah.
right? And it was like a bolt of lightning came out of the cloud, bam! And I sat up and I went, wow, this makes sense. And this is the future of medicine. So I was like a religious conversion. So I always believed that stress theory would one day, um, be, with what they were missing, as he explained it beautifully. He said, we've got this theory that there's just this one mechanism and everything, but we need a testable mechanism. We've got to have a testable mechanism and we know what to call it already. It's called the stress mechanism. And but we haven't the foggiest idea of what it looks like or where to look. And so what happened was right about that time that I went to medical school, it was being abandoned because for 30 years it had been the prevailing paradigm of medical research. And but nobody could find any clue of any testable anything that could explain any aspect of Cellier's ideas. Hans Cellier was the guy who is credited with this theory, stress theory. And and so I thought, well, this is fascinating, but I'll never have anything to do with this
Yeah.
because I want to go out, make money, chase women, whatever I'm going to do. But I I mean, I always wanted to contribute something to science, but I never dreamed that anything like this would happen to me. There I am, 10, 15 years later, I go to this conference and I get this news and I got so curious about factor eight. Now, factor eight, you know all the history about that, you guys, about
Brad, do you know the
the
No,
history of factor eight?
no, definitely not.
the Russian Revolution, its role in the Russian Revolution.
Mm-mm.
You're aware of any of that?
Mm-mm.
No, go for it.
Mm.
Tsar of Russia, um, Nicholas II, he married one of Queen Victoria's daughters, and they were carriers of this defect that doesn't affect females, doesn't affect women, but it affects sons. Okay, it's a sex-linked genetic, defect that you inherit.
Oh, and the little
Uh,
boy he had was always sick, right? Yeah,
Hemophilia.
yeah.
Had
Yeah.
true hemophilia, not Christmas disease, but true hemophilia. Christmas disease is a defect in factor nine, but um, hemophilia is a defect in the male. See, the male Y chromosome that determines sex, determines maleness, if you will. It's
Yep.
like missing a leg. So it doesn't have a backup gene. Okay. And so if you get that bad gene, you're in trouble. Because you don't have the women have the second leg they have an X instead of just that Y that's lost its tail. And am I making a sense for you guys? This
Mhm. Yeah.
anyway, so uh this little boy was born and she'd had all these miscarriages
Mhm.
but she'd produced like I think five daughters and they were just really really frustrated because they needed a son to take
That's
over.
probably her body. There was some kind of pushback for having sons. It was doing everything it could to produce daughters because it knew what it had. Interesting. That's
It
just funny.
could be. I mean, I think that's beyond the scope of my knowledge.
Yeah.
But
That's Steve Scott's knowledge. He knows all about
well,
that.
Yeah.
all the boy pregnancies would just fizzle and die.
Yeah. Yeah.
But anyway, this one fellow made it.
Yeah.
His name was Alexei.
He ended up getting shot by communists. So yeah.
Yeah,
Yeah.
there you go. See, you do know
Didn't work.
about
Yeah.
that. Yeah.
So who
Uh,
is von Willebrand and what does that have to do with that factor?
oh, well, see,
I think
that's what
probably
I
one of the
learned.
interesting stories that you've told me about is the survivors of the earthquake in Japan and how the sympathetic stress increased their von Willebrand factor and combining with factor eight and creating a
Right, right,
insoluble fibrin and
right. Well,
blocking
von
up the capillary
Willebrand
gates and whatnot. And
Yeah, von Willebrand factor is a gigantic molecule that's manufactured by the vascular endothelial cells. I mean, now the vascular endothelium is this delicate layer of cells, one cell thick, that lines all your blood vessels, and it's the sole cellular component of capillaries. These are highly specialized cells, and they do several things. Basically, from the viewpoint of stress mechanism, there's two main things they do. One is they isolate extravascular tissues from the enzymes in flowing blood. Specifically, they isolate tissue factor in extravascular tissues from factor 7 in flowing blood. Tissue factor, when it gets into flowing blood, it connects to factor 7, which is an enzyme, but it cannot exert its enzymatic properties unless it binds to tissue factor. But when it binds to tissue factor, it becomes activated. Now, same thing with von Willebrand factor. The vascular endothelium is heavily innervated in your organs, in your chest and your brain, okay, your internal organs. Your muscles and peripheral tissues are not innervated with autonomic nerve endings like that, okay? That's just for your internal organs. So, in the internal organs, sympathetic nervous activity stimulates the vascular endothelium to release von Willebrand factor into blood. And the von Willebrand factor is a gigantic molecule which binds to this factor 8C enzyme that's produced by the liver. And it's also a gigantic molecule. So then you have this humongous molecular complex. It's so big that it cannot escape blood ordinarily. It cannot pass through the vascular endothelium. So the vascular endothelium is a selectively permeable layer of, and it allows like various substances to pass back and forth. It allows factor 7, factor 10, and soluble fibrin, which is like a fibrillar protein, submicroscopic fibrillar protein, to leak through the vascular endothelium. The vascular endothelium has, the cells have muscle fibers. All eukaryotic cells have muscle fibers, but inflammation, when the stress mechanism generates thrombin, which energizes cellular activity. So when the vascular endothelial cells are exposed to thrombin, it energizes those muscle fibers, and they contract and they open up little gaps in between the cells. And so then your soluble fibrin can, and your factors 10 and 7 can escape through those gaps into the extravascular tissues. And then that you'll see when you read the book that instrumental in the wound healing process, the tissue repair process.
Right.
So, that's what the stress mechanism does is it regulates hemodynamic physiology, and by doing that, it regulates organ function because blood flow determines organ function. Okay.
So if obesity is a disease, Brad, how does what he just said connect with what you're looking at the cellular level for that obesity? I'm just
Well,
really
it's like I say, I mean, I think these things are all typically positive feedback loops, right?
Hmm.
That they're self-reinforcing systems, and if it is the biological imperative of the animal to fatten up to get through
Hmm.
winter, or sometimes this happens. There's a
that's literally a stress mechanism. It's cold. It's winter.
yeah, it's
It's
cold. It's
dark.
cold, but there's also a hibernating um, primate that lives in the tropics that hibernates through the dry season, right?
Who's
So it
that?
doesn't have to be. So it doesn't. The fat-tailed dwarf lemur.
Oh my God.
So that
Interesting.
so it's not always triggered by cold, but it is. But drought is another type of stress, of course, and they have their own system to control it. But the point is, yeah, the right, the animal is taking in signals from its environment, day length, and all kinds of what types of foods are available, and that's a whole other discussion for another day. But if the animal has decided that it something bad is coming, then it's the imperative of that animal to, okay, we better pack on some extra pounds, right? And so these systems, I believe, become self-reinforcing. And what you're saying, the stress is maybe starting to constrict the blood flow. That is, that's causing these other mechanisms to kick in. That's causing a little bit of increase in HIF1 proteins, and they're activating the things that start the glycolytic switch, and then and like I say, then the fat cells start to build, and that's putting pressure on the blood
Right.
vessels, and it all just kind of
Mhm.
snowballs together, and
And
you
th-
can't really,
Yeah.
right, you can't separate the systems. It's all part of one system. It's just
Yeah.
there's intracellular parts and there's extracellular parts.
and
Well,
this rem-
one of the things that again, I mean, like you guys are all young enough to be my kids, you know. I'm like an old fart. So one of the most amazing things for me is that since I finished my training and education and all that, um, there's been this fantastic increase in knowledge of the capabilities of the eukaryotic cell. I mean,
Yeah.
I've been astonished by this, by the research, like what Brad's talking about here.
Mhm.
There's a whole bunch of other stuff like this. These guys beavering away, doing all these elegant studies, you know. Now, I haven't focused on this, but I'm kind of peripherally aware of it, and I'm interested in all of it, but tangled up with this stress mechanism, so I've just focused on that for the last, you know, gosh, I've been working. The book like took 20 years to write, you
And this
know.
reminds me
Yeah.
also, I'm just putting the pieces together that, Ray Peat was against the traditional notion. He thought it was tenuously based in science, is the notion that omega-6 and omega-3 are essential fatty acids. And he talks about these studies where they depleted rodents of these molecules, and they would take these rodents and beat them on the backs of furniture. I don't know what kind of ethics of this study was, but apparently they didn't feel any pain. They didn't feel any pain or anything. It was like the pain was going away. And I thought to myself, well, man, that's interesting. That what if Lewis Coleman is onto the stress mechanism and Ray Peat was affirmative of that path, and it's got something to do, I think, maybe with these essential fatty acids to some degree, at least, because it's interesting how when you depleted them, they did not feel any pain, and they would whack these rodents, grab them by the tail, and smack them onto the backs of furniture. Steve, you probably do studies like that with your CO2 tank, but you know,
Well,
Well,
you're not controlling your
yeah,
variables very well
yeah.
that
I
yeah.
way.
don't know, but they did some kind of study there,
What if
and
I what if I can jump in here, David? So this is what you're saying is precisely correct. And the recent series of articles in my blog, I talk about these connections, right? So inflammatory cells, right? Uh Inflammation is fundamentally anabolic, right? If a macrophage senses that there's an invader, a bacteria, or what have you, the macrophage becomes glycolytic, and it does this even despite the fact that oxygen levels are okay, but it clicks on this product of process of glycolysis, and it's doing that to create, right. It has to rapidly build. It needs to build membranes to engulf the bacteria. It uses this system where it's producing superoxide, which is a reactive oxygen species, and they have these enzymes on the outside of the macrophages that just blast the bacteria with these reactive oxygen species, the superoxide. That's another thing that neutralizes them, but they have to go into this fundamental um, anabolic state to do this. And if you limit the enzymes that control the desaturases, the delta 6 desaturase and the delta 5 desaturase, what happens is you get much less of what they call the respiratory burst. You if the cell, because the problem, like I said, is that if you're very anabolic and you're doing glycolysis, what you need to do is you need to get rid of the electrons on that NADH, or you can't keep doing glycolysis. And so the linoleic acid is there to remove that NADH, right? And so that inflammatory process doesn't go as well or as long if you don't have the linoleic acid to continually get rid of that to continually get rid of that NADH. And as I said, then when you look at an active site of inflammation, it very quickly becomes very hypoxic, because even though the cells are doing glycolysis, which doesn't require oxygen, uh, they're using, you see the amount of those oxidized lipids in that area surge, and to make those oxidized lipids requires a tremendous amount of oxygen, but they're doing it because they need somewhere for that those electrons on that NADH to go, and it's driving forward this whole pathway of we need to get anabolic. That means we need to do glycolysis. That means we need to ferment these PUFA to remain glycolytic, and that gives us this power to build and to spray out,
Well, it enables the cell to
reactive
survive
species.
for a while in the absence of oxygen
Right.
or in a relative hypoxic state or
Right, but that but
state
that
of,
process but that process is also driving the hypoxia in that case,
uh-huh,
because it's taking oxygen. They're using oxygen to ferment just like I say, to ferment that linoleic acid. All of those pro-inflammatory, if you will, oxidized lipids that you see at the site of the infection. Well, it's getting hypoxic because they're fermenting, right? They're fermenting that PUFA to get rid of the NADH, or to
uh-huh.
remove that,
Eh,
to convert the NADH back to NAD plus, and that allows them to keep doing glycolysis. But that process requires oxygen, right? And they're not burning oxygen in their mitochondria because HIF1 prevents that, and the other things that are activated prevent that.
Uh-huh.
The reason what's burning the oxygen is the fermentation of these polyunsaturated fats, and also the fact that um, the oxygen molecules are being converted to superoxide by the enzyme systems, right? And so they're flipping the script on oxygen usage, and one of the ways that they do that is by fermenting linoleic and linolenic acid,
Yeah,
which of course is being steadily replenished as free fatty acids as they fight this battle, right? They're pulling the glucose and they're pulling the PUFA from the bloodstream to continue this process of glycolysis.
well, this is all relevant. Yeah. I want to just pitch this at you as a spitball on the side because you know there's the oh shoot, what am I blanking? The Cambrian,
Yeah.
that's a
Cambrian
big mystery.
explosion.
Yeah.
Yes.
And of course everybody's heard of that. Um, well, they've discovered these organisms thriving, hundreds of them, and every time they go down to in these deep dive contraptions, um, the ones that work and don't kill everybody anyway, in these in these volcanic rift zones. And they're finding all these tube worms and what looks like crabs and fish and you know clams, you name it. I mean, this is like hundreds of different organisms, and the microbes are so thick that they cover the rocks. I mean, it's just like a layer of both eukaryotic cells and bacteria, and I'm sure archaea, and they're eating these toxic volcanic chemicals that are just flowing up out of there, okay, in these rift zones that ring the earth like the seams of a baseball. And so these things look like familiar creatures, but
Right.
they're using different metabolic pathways, completely different from what from your land-based, terrestrial or even ocean-going marine creatures and single-celled creatures as well as multicellular creatures that we're familiar with, right? This has just been discovered very recently.
Yeah.
Yeah,
Well,
you know what I'm talking about.
to put that into perspective, if you believe the latest earliest animal fossils, or potential animal fossils that have been found are about 900 million years ago, what looks like the material of sponges in they're not very well preserved. You can't tell that it's a sponge, but
Right,
you're like, where else did this fiber come that's in this rock if it's not from a sponge?
right.
And so we could, I think we can say that animal life started around 1 billion years ago. And yet remember that back then, there was much, much, much less oxygen in the oceans than there are today. And so that first, um, the first, the hundreds of millions of years before the Cambrian explosion happened in an environment that's much lower in oxygen than the environment that we have today. My supposition being that those strange looking things um, living down deep in these thermal beds and low oxygen areas probably have been that way the whole time. And in the rest of the ocean, the oxygen levels increased, and then on the land, the oxygen levels increased, and we've moved around. But um, this is another thing I talk about on in the latest blog. So I have a post called "Pregnancy is Hypoxic," and so if you look
That's
in
true. Yeah, that's
if
true.
you if you look in the oxygen pressure in the body is similar to um I'm pretty sure it's similar to like the ocean, the deep ocean, or at 5%. I don't know exactly where that happens in the ocean, but it's a more primordial state, the low oxygen state. And that's how, of course, all cells evolved, right? As cells evolved in the absence of oxygen, and then later oxygen came, um and the cells use that to their advantage
Right.
ultimately. At first, it was probably very bad for most of them, but ultimately, some learned how to use it as a tool, right? And so when a when an egg is fertilized, one of the first things that happen within the first couple of days is in the mother, the artery that feeds into the uterus is blocked, and so the partial pressure of oxygen, it becomes quite hypoxic. And I'm arguing that that hypoxia favors a glycolytic state, a glycolytic, very anabolic state. And in those first weeks of pregnancy, those cells are rapidly dividing, and that's what you see in anabolic cells. The cancer cells are glycolytic, and they're rapidly growing. The immune cells are glycolytic, and they're very anabolic. The obese, the adipose tissues in the obese are glycolytic, and therefore presumably anabolic, and that's probably why the fat cells are growing. Right. That is the fundamental growthy anabolic state is hypoxia, and so the mother wants that wants the egg to grow, the fetus to grow, and so she restricts oxygen to keep it in a replicative state. And in the early embryo, almost all of the cells are still stem cells. They're all able to grow and divide. And then as the baby comes closer to term, more and more oxygen is can re-enter the placenta. So over time, the oxygen levels get higher and higher. And what does that do? That stimulates mitochondrial growth. That stimulates the production of reactive oxygen species in the cells, and those reactive oxygen species become the signal to the cells that, okay, we don't need to be stem cells anymore. We can differentiate, and we can become grown-up adult cells because oxygen is here, and now we're sensing the oxygen via these ROS, sensing molecules, and now we can become adult differentiated cells that are no longer just growing and reproducing.
Yeah, I'm
And
curious. I'm curious, Brad. What exactly is your research specialty?
well, I, you know, so I'm develop right now. I'm working on like cell culture, I would say. So right now I'm working on a cell culture assay that is going to look fundamentally at how changes in the environment, meaning in this case it's the cell culture medium, but of course that's supposed to represent the serum of the blood, right? When you change that, how does it affect the NADPH and the NADH and the ROS levels in the cell, and whether they're more likely to store lipids or give off lipids, and how does that process look? And so I'm sort of in the process of developing that whole thing, but that's my big research um project that I've been chatting with David about this too. So this is going to be very right. So now we finally live in an era where we have I have robots that do the pipetting. I have a very powerful microscope that can image 384 well plates. So you have 384 little experiments in a plate this big. and then we can start to use AI to build models about what's happening and what's essentially controlling the flow of electrons through the cells. The theme of the lab is follow the electrons.
Hmm. Hmm.
So that's kind of what I'm working on, and yeah, that's where my brain is these days. I've done other things. I've jumped around. I was out of science for a little while. I actually started a local meats butcher shop and pastured pork farm. So I'm
How'd you get
not
out of it and get back into it?
well, it's it's been a journey. People who relate. So essentially the long story short, that business just wasn't financially successful. I left. That's when I started writing, and the blog took off, and I've been able to get work in the lab, and I've been able to through my mostly through my supplement sales, I've been able to get a little bit of an egg together that I've been able to purchase some of this equipment, and now we're in the kind of
Wait,
starting to get into the fundraising
are
stages.
you
And
at a university or something? You must be.
no, I'm at a funky old lab building in Buffalo, New York, and it's great. It's inexpensive space, and it's legitimate lab space. It's a little out of date, but that's okay. We can live with that. I
Uh-huh.
kind of like it. I like it. It's kind of retro.
Yeah.
I mean,
I,
Mhm.
you can see these old bookshelves. They're great.
I want to ask Steve to kind of help with the bigger picture for the audience. We've got a few things here. Brad saying that, like, this is a hypoxic state that this obesity disease is in, and it's similar to how the pregnant mother creates a hypoxic state to create growth rapidly. And then we think about obesity and its association with cancer, right? Which also has to do with hypoxia. We see how
Mhm.
it's related to these similar diseases that all kind of come about from this stress mechanism. So we're putting all the pieces together. How does your understanding of oxygen and carbon dioxide and maybe some of the applications and things that you've looked at for humans, how would those come in play to help us understand how to heal or treat this problem of obesity and cancer and all the related diseases that seem to be kind of stemming from this same problem?
Yeah,
Steve
that's a good
can,
question.
yeah, yeah. Go ahead.
Yeah, a lot of this stuff that I'm hearing from Brad is very new to me, so I don't know. I'd have to read more of his blog post to really like
Mhm.
see how CO2 would play a role. I know that transdermal CO2 or injecting CO2 into fatty tissue seems to increase the metabolism and burn off fat. Um, I know CO2 will help increase angiogenesis and increase blood flow to the hypoxic uh, fat tissues and increase capillary density within those tissues and potentially help.
One moment, Brad. You said that it's a pseudo hypoxia that obesity cells might have because they're are the right because it's the oxygen's there, but it's not able to be used. Does your what you just said about oxygen going to the tissues of fat cells because of CO2, does that overcome that or no? Anybody know here? I just want to know.
Well, my understanding, what Steve's talking about is the idea that the CO2 injected into the fat tissue is allowing essentially more oxygen to get into the tissue because it's the mechanism that Lewis pointed out of the CO2 competing with the hemoglobin to deliver more um, oxygen to that tissue. I believe that's correct, Steve. And if
Mhm.
so, I think that's what was my understanding. I thought we were if that's the case, then we are sort of largely in agreement. But I don't know. Steve can tell us if that's what.
Yeah, that's my understanding of it too. I just don't know if that theory fits within what you're talking about. I need to do more research on what you're looking into. And
Steve, what about well, first of all, I remember you told me about your girlfriend who got enthusiastic about, spending a lot of time inside your CO2 suits and
Mhm.
she melted the fat off her backside. Remember
Yeah,
that?
reduce cellulite and help reduce fat just being in the suit for
This
hours at a
is
time.
is
And
is cellulite the same as obesity here, Brad, or no? Is that the same thing?
well, I
I'm
guess it
not
wouldn't
sure what.
be.
Yeah.
Yeah.
I've never quite
But
understood the meaning of that word, to be honest with you.
What, cellulite? Yeah.
Cellulite, yeah.
well,
Um,
it's kind of lumpy fat on your on
Yeah, if you look at the old paintings, people didn't have, when they were obese, they didn't have cellulite as much, it seems like.
right,
And some
right,
people have
right,
suggested it's because of the introduction of these seed oil foods.
right. Sure, sure.
Hmm.
Yeah, and I mean, again, if the CO2 is diffusing in through the skin and that's allowing more oxygen delivery to those tissues, that's exactly what I would expect.
Mhm.
I think the one thing, so I think one of the reasons that I like biology and then I think I'm pretty good at it is um I'm sort of comfortable not assigning absolutes to anything, and that everything is sort of like biology is generally counterintuitive. It's often not what you think on the first glance. It tricks you, and it's you say, oh well, just because the obese cells are now glycolytic uh, and because that process of limiting oxygen in some contexts cause the growth of blood cells does not mean that the now hypoxic adipose tissue is now fully
Hmm.
vascularized. It doesn't,
Well,
because it depends on the context. All of these mechanisms are context dependent. You
Hmm.
have to think about the individual tissue and the individual case and what happened earlier and what happened now that the obesity is developed and what was the beginning and what was the end and what
yeah.
so you can't, there's no absolutes
Hmm.
here,
Well,
And
the
I'm
but
other
as a
assuming these
general.
obesity cells are more normally the pH gradient in cells in the external environment of the cell that's a little more acidic inside of the cell and more alkaline and outside of the cell. But I'm assuming in this obesity situation or diabetes, I know in diabetes for sure that the pH gradient is somewhat reversed or the gradient is less. So the inside of the cell is more alkaline and the outside is more acidic. And that might just the external environment of the cell being acidic and have a lot of like lactate might exacerbate the situation, making a more glycolytic
Yeah,
situation. The CO2 might help reverse that pH gradient potentially, and it slightly acidifies the cells, the proteins in the cells, and helping to structure the
right.
water and whatnot, and
Yeah,
might
that's
help.
interesting because yeah, I mean the obese glycolytic cells are definitely giving off more lactic acid.
Hmm.
That's what you do. That's what you do in glycolysis, and there's evidence that as capillary blood flow goes through a adipose cell bed in obesity, that lactic acid does indeed increase across that capillary
Hmm.
bed.
And
So
I think in the obese environment, and there's less removal of the waste products too. There's less like lymphatic drainage. There's less like venous return, less blood flow. So things, these toxins, lactic's not a toxin, but these byproducts of metabolism, they tend to accumulate in the environment. And I think potentially breathing CO2, it makes you breathe faster. So you're creating like this thoracic pump almost like when you're exhaling, it's creating this positive pressure. And when you're inhaling, you're creating this negative pressure. And it's kind of drawing the lymphatics up and then pushing. It's kind of like its own heartbeat in a sense. It creates its own wave. They call it a Valsalva wave, and it might help create a better environment inside the fatty tissue where you're getting more tissue perfusion and getting more waste products removed through breathing CO2 potentially because of
Mhm.
the deeper breathing, and you're opening up everything and getting more blood flow in there. And maybe
right.
my idea is that potentially that the fatty tissue is it's kind of stagnant. A lot of those things tend to accumulate and
Steve,
poor
tell him
tissue
about
perfusion and whatnot.
the bats and the mole rats.
Yeah, I mean the mole rats and they basically naked mole rats, they generally live like an average like 34 years, where a rodent the same size normally lives like 1.5 to 2 years, maybe 3 years at the most, and they live in very high CO2 environments, and they never get cancer. They basically don't age. They don't die of old age. They die of
And
a predator or something like that. And
they're hypoxic too.
yeah, they're also hypoxic too. Yeah, so that
How
they're
does
having
that
less
fit, Brad? How can you
oxidative
be
stress and
hypoxic but yet not obesogenic or cancerous like the
Well,
naked mole
so
rat?
Wait, well, so this has been a super enlightening episode. So sure, you can be it's it's sort of like Lewis was describing before. We're looking at a photograph
Yeah.
when what's actually happening is a video is playing, right? And
Right.
so you look and you say, well, it looks hypoxic, but that might be because it has increased oxygen delivery to the tissues, is why the blood looks hypoxic because
Wow.
the oxygen is being directed into the tissues rather than hanging out in the blood, right?
Yeah.
And so though
Is that what's
I
going
don't
on with the baby in the womb? Is more oxygen getting into the cells because of
well,
the, or am I misunderstanding?
well, not necessarily, right? No, the opposite is happening, right? So what I'm saying is that there's two different, right? So in the womb, in the mole rat, if you look in the blood, it's hypoxic because the blood is circulating, like the mole rat's breathing, and as Lewis pointed out, it's probably being saturated in the lungs, even in a burrow in a high CO2 environment. But if it's high CO2, then oxygen delivery to the tissues is actually increased, right? So you have more oxygen flow to the tissues. Whereas in the hypoxia, and that causes the mole rat to be um, young, perhaps, but adult, right? It causes differentiated cells. It causes, an adult, mature organism. Whereas in the baby, the mother is restricting all oxygen flow, and so that's true hypoxia, right? Like that's hypoxic because the oxygen is limited,
Okay.
as opposed to hypoxic because oxygen delivery to tissues is increased.
How can we all be more like naked mole rats where we can live forever with no diseases?
I'm going to do an experiment on myself. Um, I I've been meaning to send it to Steve. I've got a pro or a natural gas burner that's designed to go in a greenhouse, and it's all set up to work during daylight, unfortunately, because what I wanted to do is I'm going to mount it in my bedroom, and I've got a ceiling fan in there, and so at night I'm going to close myself in with a cat and
Don't try this at home, folks.
Right.
Whatever he's about to say next. Okay.
Um, I mean, guys have been doing experiments on themselves
Yeah.
since forever, and so
Yeah.
that's what I'm doing.
Yeah.
I'm
Yeah.
not doing this to anybody else.
Yeah. I'm just teasing.
Uh, but
Yeah.
I mean, people have. I mean, they can't. The big pharma can't stop you from doing this as an individual,
Yeah.
right?
Yeah,
But
yeah,
anyway, I found this system that's all set up. Now, I don't want to use it in the summer in Bakersfield because, it's just going to jam my air conditioning bill sky high. But in the winter, it'll heat the bedroom and it'll heat the house. And
And what
so
is
that
this? What is this system?
well, it's
Do?
like a natural gas burner that, of course, when it burns natural gas, and as it does so, it generates carbon dioxide,
Okay.
and it's designed for use in a greenhouse
Well,
for seedlings and things. It doesn't take very much, just a little bit of elevation of your CO2 partial pressure, because CO2 in the ambient air is a trace gas. It's only even at sea level, it's only 0.03% of the atmospheric gas mixture. And that's not very much. And most of that hovers right at the surface of the Earth.
mhm.
And in other words, it's being produced by volcanic or bacteria way deep beneath the surface of the Earth. And it's constantly seeping up to the surface, and that's where we get our CO2. And so all it very tiny elevation of the concentration of CO2. I'll never forget my botany professor at Ohio State um, mentioned that here in this Central Valley of California, where I'm living now, back then I was at Ohio State, so he mentioned this is the most productive agricultural land in the world.
Wow.
And why is that? It's because it's surrounded by mountains. It's kind of a bowl, if you will, a valley surrounded by mountains. And the mountains trap carbon dioxide. And so it's just a little bit higher concentration of carbon dioxide here in this Central Valley of California. So everything grows the cotton grows taller and
Wow.
faster and the fruits and
Mhm.
everything. This is the most productive farmland in the world.
Wow.
Mm.
And I never forgot that. That was a practical tip. And I
Another
think,
idea you could do is if you could sleep in a tent on your bed that's
yeah,
relatively,
yeah, you
and it
could.
would accumulate the CO2 that you breathe off to some degree
Yeah,
Brad,
that
and
would,
increase the
Brad,
CO2
how does
levels
that fit
higher.
in with what you've studied with every little animal known to man, these copepods and all the things that the
Oh,
Japanese eat?
yeah, what um let's remember that when life first crawled out of the ocean, I don't know the exact numbers off the top of my head, but oxygen levels were lower than they are now, and CO2 levels were presumably higher than they are now.
oh yeah, they were way higher, way, way
Presumably
higher.
that's true, right? And so the first amphibians would have been that adapted to life out of the water would have been in a high CO2, much lower oxygen environment, and so life is not all this other stuff is this higher oxygen, lower CO2 level is much newer.
But it looks like there's
I
some kind of thing in the future that we can understand. Where we have to get these diseases under control. People can't
Yeah,
think straight. And if you can't think straight, you can't act straight, you can't do good things for people. You know what I mean? So that's it all
Right.
comes together. That's why I have Steve Scott on here. He's trying to help people.
yeah.
Steve, anything else you
Mhm.
wanted to add before we land the plane here and get some more CO2?
Yeah, just real quick. This fits into my unified theory of biology, but my understanding is that most of the oxygen we breathe comes from photosynthetic bacteria, not from leafy plants in the Amazon. Everybody gets their panties in a wad because
Yeah,
of
by the
they're
Amazon.
burning down the trees in the Amazon and everything. Oh my God, we're all going to die of oxygen starvation because of these guys. No, the vast majority of the oxygen is coming from photosynthetic bacteria that are ubiquitous on the surface of the earth and in the water too. But they take sunlight and combine it with carbon carbon dioxide, and they produce oxygen as a byproduct. Um,
The the cyanobacteria caused the oxygen catastrophe. That was the in the early days. In the early days, is the cyanobacteria were photosynthesizing, releasing oxygen, and most life on Earth perished because of the rising oxygen levels, and
Mm.
it hadn't dealt with it yet. And then eventually
yeah,
things
right.
dealt with it, and life went on.
Yeah, it caused a conversion to a new type of life forms.
Yeah.
I don't have all the answers by any means, but this is fascinating stuff. And one thing I wanted to draw your attention to, I don't have the answer to this, but the eukaryotic cells in these animals that live in the rift zones,
Yeah.
they couldn't use the Krebs cycle because that's a hypoxic environment. So they're using different metabolic pathways to generate ATP. Now, what I think happened is that all these different anatomical forms developed in these rift zones over millions and billions of years. The fish forms and the crayfish and the tube worms and clams and you name it. I mean, I don't know. There's 300, 400 of them that I've been identified so far.
Yeah.
And they look just like the stuff we're familiar with. But you bring them up to the surface, they're instantly dead. They can't use oxygen. They use the toxic volcanic chemicals. And uh, I did find a study. I blundered into a study. I don't know whether Steve sent it to me or Jeff Walden sends things to me like Steve does. And apparently, they found these eukaryotic cells that you know uh, sounded like they had engulfed some other type of organisms that with with the ability to metabolize volcanic chemicals or something. I don't know, but the theory, as I understand it today, is that the eukaryotic cell is sort of a predator cell, and that it has engulfed uh, previously free-living bacterial cells, and some of those have achieved a sort of symbiotic state within the eukaryotic cells, and the mitochondria is a good example of that. So the
Yeah, that's
mitochondria have their own uh, DNA and RNA and everything, but that's isolated from the nucleus by the thick walls of the nucleus, and I think that's to prevent contamination of the two different types of DNA. Um,
yeah,
Brad, what do
and
you think?
yeah, so the eukaryotic cell can engulf things, and sometimes they survive inside there, and then over time they lose a lot of their anatomical characteristics, but it's like a symbiotic relationship.
yeah.
The the mitochondria are doing something for the eukaryotic cell, enabling it. So what I'm hypothesizing here, and this is not in the book, okay? I'm trying to tell you that when you read the book, my thinking hadn't advanced to this point. I wondered how this happened because obviously, if uh, life progresses by evolving new stress mechanisms that adapt to unique sets of environmental circumstances. And that's the theory, right?
Right.
Uh, then the cells, um, they have to, um, you have you have intracellular stress mechanisms. In other words, each cell has a stress mechanism within itself that maintains structure and works its metabolic activity and keeps it alive. Okay, but then I'm talking about an extracellular stress mechanism that is operating outside the cells, but providing the cells with ATP and creating an internal environment in the animal or plant that facilitates the growth and survival and activity of the cells, right? They have to have oxygen, they have to have food substances, they have to be maintained in within certain temperature ranges, and things like this. And of course, acids and bases and all that. So the stress mechanism, this extracellular stress mechanism, is maintaining our cells in a happy environment. Um, well, when there has to have been a new type of eukaryotic cell that can utilize oxygen that enabled the Cambrian explosion. Because here you have all these animal forms living in the rift zones, eating these, thriving on the volcanic chemicals, which are very energetic. I think ATP is, like a holdover or remnant. It's still used as like a universal medium of energy
Yeah,
exchange. And that's what is you guys, you're the biologist, phylogeny recapitulates the ontogeny and all that. You know,
yeah,
nature's conservative. It keeps on using the same things, as long as they're working.
absolutely.
Yeah.
100%.
So there must have been a seminal event where somehow these eukaryotic cells that were thriving in the rift zone suddenly gained the ability to survive with oxygen instead of the toxic volcanic chemicals. And
Yeah, I
then,
think that
yeah,
I think that's probably exactly right.
their DNA had somehow retained all these anatomical shapes and ability to change their sizes and everything. Um, and so one of the things that I believe from my reading is that we have each of our cells has all these different genetic blueprints and anatomical shapes and metabolic pathways are they're inactivated or quiescent, and they can be reactivated under severe extreme circumstances. And the sex mechanism keeps capturing mutations and culling the ones that are destructive and harmful and retaining the ones that might be useful at some point in the future that are at least not causing any harm. And once in a while, it's got something that can be really useful under certain circumstances. So, when you have these dramatic environmental changes, like the Earth has gone through some pretty dramatic changes. So for the zillions of years that it's been around,
Yeah.
and I want to shut up at this point because I don't want to spoil Brad's fun when he reads my chapter. I want to
Yes.
see what your reaction is when you get there. Just understand that since I wrote that and published it,
Sure.
I've gone on and I've developed some that was one example I just want you to be aware of. That is my we're getting close to the point where if my book ever starts selling and getting appreciated for what I think it is, I think this book is going to be one day regarded as the Principia of medicine, the most important advance in medical theory in the entire history of recorded history, of
Yeah.
medicine. And it's going to reform and revolutionize medicine, make it much simpler to treat cancer and, crisis and critical illnesses. Right now, it could revolutionize medicine right now, just with our present medications,
I,
machines, and
I want to land.
monitors.
I want to
Yeah.
land the plane here. Steve or Brad, do you have any final thoughts or questions, or anybody have any questions for each other
Uh,
before we go
my
on?
my only statement is hopefully I don't know when this video comes out, but the article that I mentioned about the polyunsaturated fat wasting all your oxygen should be live on fireinabottle.net near the top of the thing when this comes out, so go check it out.
Mhm.
Steve,
Okay.
what do you think? Steve, what do you
Yeah, this is a
think?
great talk. I learned a lot about. I've known Lewis Coleman for a long time and studied his work, and a lot of the things that Brad talked about today was really new to me. So I'm really excited to read his blog posts and the new ones that he's coming out with.
Yeah,
What,
we
what,
need to exchange our email addresses
Yeah,
and
yeah.
um,
I could do these kind of conversations for all day, hours and hours, but we got to get out in the sun and we got to get our CO2 retention and
Yeah.
everything
yeah,
else, right? I
yeah,
know,
that's
Yeah,
great.
absolutely.
Brad, you're about to bust out that window. I can see all the energy you have in you.
I am. I'm like,
Yeah,
it's hard for me to sit still this long.
yeah.
Yeah.
You got more energy than a whistle pig for sure, don't you? Goodness. I
Well,
appreciate you guys coming on, guys. Fireinabottle.net for Brad's blog series. We got stress.org where you can learn about the American Institute of Stress, where Dr. Lewis Coleman is their chief scientist. I'm sure that's a very stress-free work environment there, right?
oh, well, yeah. One other little plug. Um, Dan Kirsch, who runs the American Institute of Stress at this point, has financed a movie that just got completed a couple of weeks ago, and I can't. He made me swear to God I wouldn't share it with anybody because I would love to give you the link for it. But I think he wants to see if he can win some awards to draw more attention to it. Um, and so he's working on that. So he made me
Cool.
swear I wouldn't share
Yeah.
the video link unless you want to come here and visit me in Bakersfield, which is about the only thing worth seeing or doing in Bakersfield. Um,
Yes.
but as soon as the movie becomes available, I'll certainly share it with all you guys.
And Steve,
Um,
Steve, what do you want people to leave with?
Yeah, that carbon dioxide is not a waste gas. If you think about it, we don't store any oxygen in our body, even though it's essential, and we would die without it within minutes. Yet we don't store any because it's very reactive. And CO2 is we actually retain CO2. We actually blow off all the oxygen that we don't use, and CO2 is the good guy, and um, oxygen is more of a toxin, more reactive than even carbon dioxide because we store carbon dioxide and oxygen we don't. And CO2 is necessary for oxygen to be delivered to the cells. Kind of like
well,
the protective wings around oxygen to help
yeah,
deliver it and also protect you from the overproduction of the reactive oxygen species and whatnot.
David, we could do a
Yeah.
whole one of your shows just on the anesthesia hoax
Wow.
that
Yeah.
vilifies carbon dioxide as
Goodness.
toxic waste like urine.
Wow.
Um, and there's also, I think, DuPont is a major player in
Yeah.
maintaining this nonsense about carbon dioxide.
Yeah.
Trump signed an
Yeah.
executive order, that says that carbon dioxide is harmless and useful and everything.
Wow.
Of course, he's thinking about encouraging oil production
And
and
just be careful about injecting it in certain places. We don't want to get anybody harmed.
Yeah.
it's
Also, I've
pretty
been
hard.
working on a book about CO2, working on it for about
Okay.
like last year and a half, and you'll be able to get it from carbogenetics.com
Yeah,
once it's available
I want
in a
to
couple
give a
months.
shout out to Carbon Genetics because I've used their inhalation and their bodysuit, and it's a lot of fun. When I get in the bodysuit, I look like the girl from Willy Wonka after she blueberry candy, and that's why I like to take photos and say, "Hey, look, this is how good this sugar diet's doing for me." See, and I send photos.
Yeah.
What's
Yeah,
happening?
but when you get out of it though, then you look a lot better. You look like
Yeah. Oh, yeah.
Superman or
And look at that
something.
skin I got. Yeah, look at that. You wouldn't believe I'm
Yeah.
57. Can you believe it?
Yeah.
All right. You take care, guys. I appreciate your time. Remember,
right.
you can email us hello@neighborschoice.com. Leave a comment or question you have in whatever facility that you're watching this program, whether it's a social media video platform or audio platform. We'd love to hear from you. And this is what we do here, folks. We have many people have told us that we're like academia in exile. We don't have the full facilities and the funding, but we have this network of people unlike anything in the world where we can bring together a Manhattan Project for Cancer, which is something I've been doing with a whole host of people, or we do something like this where we have all these fascinating conversations about oxygen and its role in relationship to CO2 and obesity and everything else. This is cutting edge stuff, folks. I appreciate your time and thank you all for participating in it.
Control.